Usually when we talk about a medical diagnosis, there's this underlying expectation of precision, I guess. Like it feels almost like engineering.
Yeah, totally. Very mechanical.
Right. If you break your arm, the X-ray shows that jagged white line, and the doctor just points at the film and says, there it is. You look at a photograph of the damage, and the story is obvious.
It's comforting in a way, isn't it? The visual is binary. I mean, it's
Yeah.
either broken or not broken, diseased or healthy.
Exactly. But imagine if, instead of just looking at the aftermath of a disease in a static black and white photograph, you could watch the exact moment a single healthy cell goes rogue. I'm talking about a live-action movie of the very first completely invisible steps on the journey to becoming cancer, happening in real time inside a living body.
Which is just, honestly, it demands a completely different paradigm for understanding the disease. We are essentially moving out of the realm of traditional pathology, which, relies on looking at dead, chemically fixed tissue on a glass slide. And we're stepping into the realm of living, breathing, cellular ecology.
And that is exactly our mission for this deep dive. We are going to explore the earliest microscopic steps of tumorigenesis, how cancer actually begins long before a tumor ever forms.
Right.
And to do this, we are relying on a single, incredibly dense but groundbreaking source. It's a 2026 review article from Trends in Cancer, literally titled Watching Cancer Begin. And I've got our expert guide here to help us navigate this microscopic world because, honestly, the biology here is wildly complex.
It really is, but it's also incredibly elegant once you see the mechanics of it. Historically, we only ever understood cancer from static endpoints. You either had a fully formed tumor or you didn't.
You saw the end of the war, basically.
Exactly. You saw the end of the war, but never the tiny battles that led up to it. What this paper outlines is that right now, we finally have the technology to watch mutant clones survive, persist, cooperate, or get completely eliminated in real time.
Okay, let's unpack this. Because to watch these battles, we have to talk about the cameras first. The paper calls it an imaging revolution. But I have a huge practical pushback right out of the gate here.
Lay it on me.
How on earth do we actually see a single microscopic cell inside a living, pumping, moving organ? I mean, if you shine a light into a liver, doesn't all that dense flesh and blood just get in the way?
Yeah, that is the exact physics problem researchers faced for decades. The gold standard tool they use now is called intravital microscopy, or IVM, specifically utilizing high-resolution multiphoton imaging.
Okay, multiphoton imaging.
Right. But your instinct is spot on. Light scattering is a massive hurdle. If you've ever, held a flashlight up to your palm, the light just diffuses into a blurry red glow.
Yeah, you just see your hand glowing red.
Exactly. Flesh absorbs and scatters photons. Because of that, even with the most advanced lasers, you could normally only see about 100 to 200 micrometers deep into a visceral organ, which is a tiny fraction of a millimeter.
Wait, which means if the rogue cell is hiding even half a millimeter deep, you're just completely blind to it.
Completely blind. So researchers had to get creative with mechanical engineering. They developed surgically implanted imaging windows.
Imaging windows, like literal windows.
Literal windows. Imagine a microscopic glass porthole set into a tiny titanium ring, permanently installed directly into the skin or over an organ in an animal model. They've customized the shape and flexibility of these windows for the mammary gland, the intestine, the skin, and even the brain.
That is wild. It's like setting up a hidden trail camera in the forest, but the forest is a living intestine, and you can just keep checking the live feed.
That's a great way to visualize it. It allows researchers to return to the exact same microscopic field of tissue down to the exact same cluster of cells over days, weeks, or even months.
Wow.
And for areas where a simple window isn't enough, like deep inside the brain, they've developed a tool called Deep 3P.
Okay, Deep 3P. How does that cut through brain tissue? That sounds impossible.
It's fascinating. It uses 3-photon microscopy combined with adaptive optics. And adaptive optics is actually the exact same technology that astronomers use on massive telescopes.
Wait, like looking at stars?
Yeah. They use it to remove the atmospheric twinkle from stars so they can get a clear image of a distant galaxy. They apply those exact same algorithmic corrections to the distortion caused by living brain tissue.
That is insane.
Right, and they pair that with artificial intelligence denoising, which basically separates the background static of blood vessels from the actual cells. With Deep 3P, they can see past 1 full millimeter deep into a living brain without using any chemical labels at all.
Okay, so we have the cameras in the portholes. But here's the next problem I see. A brand new mutant cell looks identical to the billions of healthy cells around it. You can't just look through the window and spot the villain.
No, you definitely cannot.
Right. We need a way to put, like, a high-vis neon jacket on a single mutant cell so we can actually track it. The paper mentions lineage tracing. How does that actually work mechanically?
So it relies on genetically engineered mouse models. Scientists use sophisticated genetic systems with names like confetti, polylox, or the DRAG model. The way it works is essentially a genetic trigger system, often called CRELOX. Think of it as molecular scissors paired with a stop sign.
Okay, scissors and a stop sign. Walk me through that.
Sure. So inside the DNA of these engineered cells, there is a gene for a bright fluorescent color, say neon green. But right in front of that color gene is a genetic stop sign that prevents the cell from glowing.
Okay, I'm following.
When researchers want to start the experiment, they inject a very specific trigger chemical. This chemical activates the molecular scissors, which then snip out the stop sign in just one specific cell.
Oh. And once the stop sign is cut out, the neon green light turns on permanently.
Precisely. And because that change is literally cut into the DNA, when that one tag cell divides into two, both of the child cells inherit the missing stop sign. They both glow green, then 4 glow green, then 8. You can watch an entire family tree, a clone of cells, expand over time.
That makes total sense. That explains how we know where the cells are. But the paper also talks about live cell biosensors. And reading through that part, it sounded less like a GPS tracker and more like a molecular mood ring.
I love the mood ring analogy, but let's look at the physics of how it actually changes color, because it's brilliant. The mechanism is called FROT, first resonance energy transfer. Biosensors don't just show location, they show what the cell is actually doing internally.
Okay, how?
Imagine you have 2 proteins inside a cell, and you attach a blue glow stick to one and a yellow glow stick to the other.
Okay, 2 proteins floating around, glowing different colors.
Right. Now, when a specific cancer-driving pathway activates, say, a pathway called PI3K or RAC1, it causes those 2 proteins to physically bind together. When they get extremely close, the physics of FROT takes over. The energy from the blue glow stick literally transfers into the yellow glow stick, causing the whole complex to suddenly emit a bright green light.
So, by looking through the microscope, if you see the cell shift from blue to green, you know the cancer pathway just physically clicked on in real time.
Exactly. You can actually watch a single cell react to its environment, or even watch its internal pathways shut down when you administer a targeted cancer drug. They have another one called KIKGR, where a protein permanently changes from green to red when hit with violet light, letting scientists permanently tag a cell's current state.
That is so cool.
And they have other visual tools too, like the Fuji system, which is essentially a traffic light for cell division.
A traffic light?
Yeah. The cell's nucleus glows red when it is resting in the G1 phase. But the moment it begins copying its DNA to divide, the internal proteins shift and the nucleus turns green. Through the imaging window, you can instantly see an entire field of cells and know exactly which ones are dormant and which ones are rapidly multiplying just by looking at the red and green lights.
Wow. Okay, so if you're listening to this right now, let's synthesize what we just learned. We now have the technology to surgically implant tiny glass windows into living organs. We can use genetic scissors to permanently tag a single rogue cell neon green, and we can use molecular mood rings like the Fuji system to watch the real-time chemistry happening inside that cell as it divides.
It's an incredible toolkit.
It really is. I mean, think about your own body for a second. If you had a microscopic window into your tissues right now, what kind of hidden, colorful cellular dramas do you think you'd see playing out?
It's a profound thing to visualize. But there is a massive limitation to these imaging windows. They're heavily localized. It's like watching a high-definition security camera pointing at one specific hallway. It's fantastic for the hallway, but what if you need to understand the architecture of the entire building?
Right, because if you're only looking at a fraction of a millimeter, you might just be looking at the wrong spot while the cancer starts next door. How do we move from a zoomed-in video to a map of the whole landscape?
This is where spatial transcriptomics and 3D tissue clearing come in. Historically, when doctors or researchers look at tissue, they use what are called tissue microarrays, or TMAs. These are 2-dimensional slices of tissue shaved incredibly thin, maybe 5 micrometers thick.
And the problem there is obvious. If you have an entire loaf of bread with exactly 1 raisin baked into it, taking a single paper-thin slice gives you a nearly 0% chance of finding the raisin.
Exactly. You miss the bad neighborhood entirely. Early mutant clones are incredibly rare and scattered. So instead of slicing the tissue, researchers now use 3D tissue clearing.
How does that work?
They take an entire intact organ, and they treat it with special chemical solvents. These solvents selectively dissolve and wash away all the opaque lipids and fats, the stuff that scatters light, while leaving the structural proteins and the tagged mutant cells perfectly intact.
So the whole organ literally becomes transparent, like a glass liver.
It looks exactly like glass. Then they can use lasers to scan the entire 3D volume, hunting through the complete organ to find every single scattered mutant clone.
Okay, so we have the cameras, we have the tags, and we have the transparent 3D maps. Let's actually look at the cells themselves. This is what I was most eager to understand.
Let's get into it.
What happens the moment a cell gets a dangerous cancer mutation? Because the way we talk about it in pop culture, the second a cell mutates, it turns into a supervillain and starts rapidly growing.
What's fascinating here is that narrative is almost entirely false. When a cell gets a severe cancer mutation, the vast majority of the time, it simply dies. It gets completely washed away.
Wait, really? The body just gets rid of it?
It comes down to the mechanical concept of tissue hierarchy. Think of your epithelial tissue, like the lining of your gut or your milk ducts, as a biological conveyor belt.
Okay, I'm picturing a factory line.
Right. The engine driving that conveyor belt is a long-lived stem cell sitting at the very bottom in a little pocket called a crypt. That stem cell constantly divides, producing specialized worker cells. These worker cells ride the conveyor belt up to the surface, do their job absorbing nutrients or secreting milk for a few days, and then they naturally die and are shed off the surface to make room for the next batch.
So if one of those worker cells halfway up the belt gets hit with a blast of radiation and sustains a terrifying cancer mutation, it just doesn't matter.
Most of the time, no. It just keeps riding the belt until it reaches the edge, falls off, and is destroyed.
That's amazing.
The paper highlights a brilliant study where researchers sporadically deleted BRCA1 and TRP53 in the mammary gland. Now, those are two of the most famous, most dangerous tumor suppressor genes in human biology.
Right. If you have those mutations, you're at a huge risk.
Exactly. When you lose them, you are highly prone to cancer. Yet, in the study, about 90% of the cells that received those severe mutations were eliminated almost immediately.
Because they were just worker cells on the conveyor belt. They got mutated, but the belt kept moving, and they were naturally shed before they could ever multiply into a tumor.
Exactly. The normal daily physical turnover of your tissue is a massive built-in safeguard. For a mutation to actually survive and become fixed in the tissue, it usually must strike the long-lived stem cell at the bottom. The engine itself has to break.
Let's pause and lock that in because that is incredibly reassuring. So here's a quick recap for the listener. 2D tissue slices often miss the rare mutated cells, but treating organs with chemicals to make them look like transparent glass gives us the whole 3D map.
Yep.
And crucially, we now know that getting a cancer mutation isn't a death sentence for a cell because 90% of them happen in short-lived worker cells that are naturally shed. Does it change how you view cancer, knowing your body is likely clearing out heavily mutated cells every single day just through normal wear and tear?
It is a beautiful defensive system, but it does leave us with the darker scenario.
Right. What if the mutation does manage to land in the engine? What if it hits the stem cell at the base of the conveyor belt? Does it immediately explode into a tumor then?
Still, no. Because stem cells don't live in isolation. There isn't just one engine. There is a pool of stem cells packed tightly together at the base of the crypt, and they are constantly fighting each other for physical space and survival signals.
Fighting each other.
The paper describes this beautifully in the intestine. It's essentially the Hunger Games of the gut.
The Hunger Games, meaning they literally kill each other.
In a way, yes. Stem cells are constantly replacing one another. If a mutant stem cell wants to survive and expand, it has to physically displace its healthy neighbors. To win this spatial competition, some mutations turn cells into what the literature calls super competitors.
Okay, give me a mechanical example of how a super competitor wins. Do they just divide twice as fast?
That's the logical assumption, right?
Yeah.
But biology is more devious than that. The classic example involves a mutation in the APA gene, which is a major early driver of colon cancer.
Okay.
When a stem cell gets an APA mutation, it doesn't actually speed up its own growth. Instead, it rewires its internal machinery to secrete a lipid called NOTAM.
NOTAM. What does NOTAM do?
NOTAM is a chemical antagonist. Specifically, it blocks something called WNT signaling. WNT is the essential growth factor that tells the stem cell, hey, you are a stem cell, keep dividing.
Oh, I see where this is going.
Yeah. The APA mutant secreting notum basically neutralizes the WNT signals reaching the healthy neighboring cells. Starved of their survival signal, those healthy stem cells get confused, differentiate into worker cells, and ride the conveyor belt away to die.
Oh, wow. So if this is a foot race, the epic mutant isn't running faster at all. It's just actively tripping the runners next to it so it can take their lane and cross the finish line by default.
Pure chemical sabotage. And if we connect this microscopic sabotage to the bigger picture, these turf wars are heavily influenced by the macroscopic forces of your whole body.
You mean systemic forces change the rules of the microscopic Hunger Games? How?
Take diet, for example. The paper outlined how a high-fat diet physically alters the intestinal crypts. The excess lipid nutrients actually expand the physical size of the stem cell pool. By increasing the sheer number of competing stem cells in the arena, the mathematical odds shift. It creates a larger, more permissive environment where a mutant cell has a higher probability of slipping by or establishing dominance.
So a high-fat diet literally changes the architecture of the arena to favor the mutants.
Yes. And hormones play a huge role, too.
Yeah.
In the mammary gland, the natural estrous cycle, the hormonal waves that drive the menstrual cycle, causes the epithelial tissue to constantly expand and contract.
Right, the tissue remodels itself.
Exactly. Huge waves of cells proliferate, and then huge waves die off. This constant physical remodeling creates literal spatial vacancies. Mutant clones use those vacancies to expand their territory over time, moving into the empty gaps left by dying healthy cells.
The whole body is pulling the strings on these tiny cellular battles. Okay. Quick synthesis for the listener. Mutant cells like APC use chemical weapons, secreting notum to starve and kill their healthy neighbors. And whole body factors like a high-fat diet expanding the stem cell pool or hormone cycles creating physical empty spaces directly rig the odds in these microscopic turf wars.
That's the takeaway.
So how does knowing that your daily diet changes the literal Hunger Games happening between your microscopic stem cells make you think about your next meal? It's pretty intense.
It certainly makes you respect the systemic connection. But the consequence of those mutants winning the space brings up something truly shocking.
Yes. Here's where it gets really interesting and, frankly, deeply unsettling. The paper drops a statistic that made me stop reading and just stare at the wall.
I know the one.
It states that 3% of normal, healthy-looking human breast epithelial cells in all women are aneuploid, meaning they have abnormal mutant chromosomes, and they have successfully won the Hunger Games and undergone clonal expansion. But if you look at a mammogram or even look at the tissue under a microscope, it looks perfectly normal.
This is a phenomenon known as field cancerization. For a long time, we viewed cancer as a strictly localized event, a single bad spot. But deep genetic sequencing reveals that huge, sweeping fields of morphologically normal tissue can actually be composed almost entirely of mutant clones.
They are like massive sleeper cell networks just hiding in plain sight. But how are they not forming tumors if they have the mutations?
Because they are trapped.
Yeah.
They have the dangerous mutations, yes, but they are perfectly constrained by the physical, structural architecture of the tissue.
Caged in.
Exactly. The literal 3D shape of the mammary ducts acts as a physical cage that prevents the mutant cells from growing outward into a chaotic mass. They are mutants, but they are following the spatial rules of the neighborhood.
But wait, if there are entire fields of these mutant sleeper cells, that totally busts one of the biggest myths in popular science. We are always taught the lone wolf theory, that cancer starts from exactly one bad cell that goes crazy.
The paper explicitly corrects that misconception. Early lesions rarely happen in isolation. They actually rely on something called clonal cooperation.
Teamwork. Cancer cells use teamwork.
Unfortunately, yes. Researchers discovered that if you have adjacent sleeper clones that harbor different mutations, say one clone has a WANT pathway mutation, another has a KRAS mutation, and a third has a MECA mutation, they can actually collaborate to form a multiclonal adenoma.
How does that even work mechanistically? Do they share nutrients or something?
They share the labor of altering the environment. Think of it like this. A cell with a KRAS mutation might not have the right tools to form a tumor on its own. But that mutation causes it to secrete factors that disrupt the local chemical gradients in the tissue. It creates a sort of chemical fog.
Okay, a fog.
That fog inadvertently protects a neighboring cell that has an AMP mutation. The AMPic mutant uses that permissive fog to finally break out of the tissue cage and initiate a lesion. They compensate for each other's biological deficiencies.
That is just diabolical. So let's wrap our heads around this section. Healthy-looking tissue can actually harbor massive fields of sleeper cell mutant clones perfectly held in check by the physical architecture of the organ. And tumors aren't always a solo act. Different mutant clones will literally secrete chemicals to create a fog that helps their neighbors break out and grow. So to you listening, if large fields of our tissue are already carrying mutations, what is the spark that finally pushes them over the edge into disease?
That question brings us to the final and perhaps most important piece of the puzzle, the microenvironment, the neighborhood outside the epithelial cells.
Okay, so we're no longer talking about the cells on the conveyor belt. We are talking about the factory floor itself.
Precisely. And the spark isn't just chemical, it is physical. Mechanical forces dictate cancer initiation. The paper highlights a study on skin cancer involving a mutation in the smoothened signaling pathway which drives basal cell carcinoma.
Okay.
Researchers found that a cell possessing this severe mutation cannot reprogram itself and start a tumor unless the extracellular matrix, the ECM, the non-living physical scaffolding that holds our cells together, provides the exact right mechanical stiffness.
Wait, stiffness, like how rigid the tissue is.
Exactly. If the scaffolding is soft and pliable, the mutant cell just sits there, harmless. But if the scaffolding stiffens due to age, inflammation, or scarring, that stiffness physically pulls on the membrane of the cell.
Oh, wow.
That mechanical tension activates intracellular forces that literally stretch the nucleus, pulling the trigger on the cancer genes. The physics of the neighborhood are required to initiate the disease.
So the wood is soaked in gasoline, but it needs the physical friction of the stiff scaffolding to actually strike the match. Are there other things in the neighborhood striking matches?
Yes, the cellular accomplices, immune cells and structural cells. In breast tissue, researchers used IVM to watch physical interactions. They found that a specific type of immune cell, a CXCR4 positive macrophage, makes direct physical contact with the mutant epithelial cells.
And let me guess, it doesn't destroy them.
No. Instead of destroying the mutant, as an immune cell should, that physical handshake actually enhances the mutant cell's ability to transform and grow.
The immune system gets tricked into being an enabler.
Indeed. And in the intestine, there are structural cells called mesenchymal fibroblasts. Their normal job is to maintain tissue framework. But near a mutant clone, these fibroblasts get hijacked and start secreting a lipid called prostaglandin E2, or PGE2.
What does this lipid do to the mutant cell?
The PGE2 lipid binds to the outside of the mutant cell and initiates a cascade that physically shoves a specific protein called YAP straight through the nuclear pores and into the center of the mutant cell's nucleus.
Okay, and YAP does what?
Once the YAP protein is inside the nucleus, it acts as a master switch, turning on all the genes required for the tumor to rapidly expand. Without the fibroblast squeezing that lipid onto the mutant cell, the YAP protein stays out of the nucleus and the tumor never initiates.
This raises an incredibly important question. If the mutant cell is completely harmless without the stiff physical scaffolding pulling on it, or the immune macrophage shaking its hand, or the fibroblast shoving proteins into its nucleus, are the mutations the real danger, or is it the permissive neighborhood that actually dictates the disease?
That is the exact paradigm shift this paper is pushing us toward. It is an ecological view of cancer rather than a strictly genetic one.
Let's summarize this final piece of the puzzle. A genetic mutation alone is rarely enough to start cancer. It requires mechanical physics, like the stiffness of the tissue scaffolding pulling on the cell membrane, and chemical accomplices from hijacked immune cells and structural fibroblasts to pull the trigger.
That's it in a nutshell.
So think about this for a second. If the neighborhood is what truly allows the criminal cell to thrive, should we be trying to fix the cell or fix the neighborhood?
It completely shatters the reductionist cell-killing view of oncology we've held for decades.
Wow. Okay, we have covered an immense amount of ground today. We've moved from looking at static, dead photos of cancer to a cinematic 3D tracking of living cells. We've learned that cancer isn't just one rogue cell deciding to divide endlessly. It's this incredibly complex ecological battle.
It really is an ecosystem.
We've got survival on the conveyor belt, the Hunger Games of spatial competition, chemical sabotage with NOTAM, teamwork between sleeper clones, and a microenvironmental neighborhood that either cages the mutant in or enables its escape. So, what does this all mean? Where does this leave the future of medicine?
It changes everything about how we approach intervention. If we can use spatial transcriptomics to map these high-risk fields of sleeper clones before they ever turn into tumors, and if we can use live imaging to understand the exact neighborhood signals that enable them, We might not need to invent new ways to hunt down and poison every single mutant cell. We might just need to alter the ecology.
Right. Change the stiffness of the extracellular matrix, block the PG2 lipid coming from the fibroblasts, stop the immune cells from shaking hands with the mutants. It's like changing the pH of the soil so a weed can't physically take root, instead of pouring toxic herbicide all over the garden hoping to kill the weed after it's grown.
Exactly. True intervention before the cancer ever truly emerges.
Which leaves us with a final, completely new thought to mull over. If mechanical physics, specifically the stiffening of our tissue's extracellular scaffolding, is what finally pulls the trigger on these mutant sleeper cells, what if the process we call aging isn't actually about our cells getting old and tired?
Well, that's an interesting thought.
What if aging is primarily just the slow degradation and stiffening of our bodily scaffolding? If our bodies are already incredibly skilled at eliminating 90% of mutant cells naturally, what if the ultimate cure for cancer isn't inventing a new toxic drug to kill tumors, but simply learning how to give our tissue's natural conveyor belt and competitive environment a slight physiological upgrade? Thank you so much for joining us on this deep dive. Keep questioning the invisible world within you.